All right, biology kiddos, we're going to talk a little bit about cellular transport. This is the last section of the introductory cells chapter. And some of the stuff here is going to be a review, but hopefully it'll be a review that will help you understand the concepts a little bit more clearly. So let's begin by talking about something called diffusion. Now, we mentioned diffusion a little bit earlier in the cell chapter, but I'm not real sure that we actually fully defined it. So what diffusion is diffusion is where we take particles from an area of high concentration to an area of low concentration. Now that's kind of the science definition. The easier definition is right here in gray. From where there's a lot of stuff to where there's not as much stuff. And so if you look at the little GIF over here on the side, basically it's just a glass of water and you drop some food coloring into it and we don't even have to touch it, okay? There's no stirring or anything going on, but what you see is the particles are dropped in at the top, really concentrated at the top, and then they spread out. And are essentially going to spread out and cover up the entire container. And if they don't really show this here, but if they left it a little bit longer, the whole glass, each glass of water would essentially become one color altogether. Now, going along with the term of diffusion is this idea of dynamic equilibrium. So let me just real briefly sort of talk a little bit about what that is. So let's say that in one area of the container we had a bunch of particles over here, okay? And let's make a dividing line. And then let's say on the other side, we only had two particles. Okay? So what dynamic equilibrium means is that the concentrations are the same no matter what. And so what's really weird about this is that it's not that nothing is moving. In fact, this stuff is always moving. That's what the dynamic part means. Dynamic means things are moving or in change. And so let's say that I took one particle over here, okay, and moved it, and then I took another particle and moved it back, okay, and that every so often that happened again, that I took a particle and moved it, and then I took another particle and moved it. And so the concentrations on either side never changed. I had 5 over here and 2 over here the whole time, even though it wasn't the same molecules the whole time. This is what we call dynamic equilibrium. It means that the flow this way and the flow this way are equal to each other. Okay, so just keep that in the back of your mind as we talk about a couple of things in the next couple of slides. And that's what's going on in diffusion. Essentially, when the cup finally gets completely yellow, it's not that particles stop moving. They're always moving. But it's just that the concentration is the same no matter what then. Okay, so we mentioned earlier the idea of facilitated diffusion. Now, what facilitated diffusion means is that diffusion would normally happen across the cell membrane, sort of like we just saw. But there are some particles that are a little bit bigger and can't as easily fit through the cell membrane. And so facilitated diffusion means that there are going to be protein channels that are going to allow that diffusion to take place, okay? So what I really want you to look at is this part right here. Whoops, hold on. Is this part over here on the side where we've got a protein channel here. Okay, this carrier protein stuff is something different that we'll talk about here in a second. But what we've got going on here is there's a protein channel here that essentially is filled with water, okay, or fluid, whatever is going on there. And so this is inside of the cell, and this is outside of the cell. And what's happening is that the diffusion then, since some of those things can't get through the phospholipid part of the cell membrane, but they can get through if there's a channel here. And so if you look over here, I've got a bunch of these green hexagons over here, not as many over here. But let's say that they couldn't make it through the cell membrane here, but what they can do is they can flow through this protein channel to be able to get through. That's the facilitated part of the diffusion, meaning that they can't really get through the membrane on their own, but if there's a channel, a pathway, then yes, they're going to equalize the concentration as much as they can by doing diffusion through that protein channel. So, you guys have probably heard this term a ton of times. Probably all the way back to middle school, maybe even elementary school, is the idea that osmosis is water moving through the membrane. And we're talking, of course, about the cell membrane in this case. So I want to talk a little bit about how water does that, because what we sometimes think is that like the stuff moves through and equalizes the concentration, but in the case of cells, what often happens is that water is going to go through to try to equalize the concentration. So let's look at a couple of situations here. If we look over here at the initial state, okay, what we've got here is we're just saying that we've got a solution on each side, a bunch of particles. Doesn't really matter what they are. We could say that they were sodiums or potassiums or whatever. Okay, we've got a bunch on this side, not as many on this side, and we've got a membrane here between them. Think of the cell membrane as essentially what we want here. Now, there's a lot over here and not much over here. So what an ordinary diffusion would say is that we want to be able to, some of these particles get through the membrane and come over here and equal things out. But that can't happen. Let's say that these particles are too big and they can't make it through the membrane. Let's say there's something like sugar molecules or proteins or something, and they're just too big to make it through the membrane on their own. But water is a pretty small molecule, and so water can go through this way. Okay? And what that will do is that once there becomes more water over here, it essentially spreads the particles out so that they're essentially even then, and there's not a bunch all bunched up concentrated here and not many here. But if we look over here on this side, the concentrations are about even. If we took like a little chunk and looked at the density of it, okay, there's about 4 in that little box, and in the same size box on the other side, there's about 4. If we did that over here, okay, in a certain box, that was a terrible box, but let's say there's 4 there, and if we did that same size box over here, we've only got 2, okay? So we've equalized the concentration by water going across the membrane, okay? So this is kind of a hard concept to grasp. Sometimes it's easy. The idea of diffusion, of stuff moving across the membrane, is pretty easy. But remember, what's happening here is that water is flowing through this way. Okay, and it's really weird because what actually ends up happening is, like, if we had a U-shaped tube, the water levels would be equal on both sides to start with, but because of the osmotic pressure, okay, the pressure of the osmosis, one side will actually end up being higher than the other side. So you see this side is higher than this side, and what this little thing here is that's a little measure of something that we call osmotic pressure. Now, we're not going to talk a ton about that this year, but where it really matters is for plants, okay? And when we get into a little bit of the stuff with plants, we'll talk a lot more about osmotic pressure. But the osmotic pressure is just the difference because the water has flowed through to equalize the concentrations. All right. So let's jump into what is undoubtedly a few sort of new words for everyone here. So we're going to talk about 3 different situations that can occur within the cell. So the first one we're going to talk about is an isotonic solution. Okay, and this is kind of the easiest one. When you see the prefix iso in something, iso means equal. Okay, or the same. Sometimes we can say, but equal is basically what it means. And so what that means is if we have an isotonic solution, then the concentrations inside of the cell and outside of the cell are the same. So just to jump back here a little bit, if we had a cell membrane, this is inside the cell, this is outside the cell, and it would basically mean that I've got the same number of hexagons on this side as I have on this side. That would be an isotonic solution. Now that is the ideal state. That's what we want for our cells. We want them to be in an isotonic solution, because then what happens is that the rate of water flow in is the same as the rate of the water flow out, and the cell basically stays in its nice healthy little shape. You see a nice healthy red blood cell here. So isotonic is sort of the ideal that we're shooting for. However, there can be a couple of different ways that this can go bad. The first one is called a hypertonic solution. Okay, so hypertonic is going to be this case over here. And what hypertonic means is that the concentration outside of the cell is higher, okay? And so if the concentration outside of the cell is higher, then the water wants to go out and equalize that pressure. It would be kind of like here, so it would be like this is the outside of the cell, and this is the inside, so the water goes out of the cell to equalize the pressure. Now, what that does, of course, is now the cell doesn't have as much water in it, and so the cells shrink up and they become ineffective, and the cells can't function the way that they should because they don't have enough water in the cytoplasm anymore, because the water has left to try to equalize the solution. And you see that the water is leaving very rapidly here. The third way that we can see this is a hypotonic solution. Okay, and a hypotonic solution means that there's a greater concentration inside of the cell. Now what happens in that case is instead of the water leaving the cell, is that we get a bunch of water trying to go into the cell. Now the cell membrane, there's only so much of the phospholipid membrane there, and so once you get too much water going into it, essentially it's going to cause the cell to blow up. And you see this little picture here of a cell lysing, a cell coming apart, because it's in a hypotonic solution. Okay, hypotonic would basically be you take a cell and you threw it in just some regular water. Well, the concentration inside of the cell is a lot higher. There's a lot of stuff inside the cell, so the water goes rushing into it, causes the cell to burst open. Okay, so we've got 3 situations there. Now, of course, what we want is isotonic. Okay, we want the concentration to be the same inside and outside of the cell. So hyper means the concentration is higher outside. Hyper means, like, think about hyperactive. Hyperactive means more active than is necessary, so that means more concentration outside of the cell. Hypo means less, and so there's less concentration outside of the cell than it should be. Iso means equal, so it should be the same. All right. Let's wrap up by talking a little bit about active transport and about one more thing after that. Active transport means that you need energy to get stuff through the cell. So everything that we talked about so far, okay, the facilitated diffusion, the regular diffusion, osmosis, all of those are what we call passive transport. They don't require any energy. But the fact of the matter is that we do need energy in most cases to get stuff to actually go through the cell. Okay, so in active transport, we actually have to have energy come in. We have to use up energy to get stuff to go through across the cell membrane. So everything we talked about so far, it happened on its own. It didn't require any extra energy. But a lot of times it does require extra energy. It's one of the reasons why we need energy in the cell. And so here's a good example of this. If we look at this diagram, the hexagons here, the orange things, they're sodium ions. The little oval things are potassium ions. And if you look at this, what you can immediately tell is that there's more sodiums outside of the cell, that's extracellular, outside, than there are inside of the cell, intracellular. Okay? And so, but what the cell wants to do is it wants to get rid of these sodium ions from inside of the cell. Now, naturally, that's not going to happen because there's already more of them outside than there are inside. So diffusion says, no way, we don't want more outside. So if to put more outside, you have to work against the concentration gradient. Now that seems like a really fancy term, but that just means we're working against the idea that there's already a bunch out here and we want to put more out there. Kind of like if we had, say, a room packed with people and it was really packed. To get more people in, we've got to squish them in to get them in there. Okay, or like you're packing for a trip and you've got clothes in there already. Well, to get more clothes on, you've got to push down really hard to get them there. And so that's basically what active transport is doing. It's pushing hard by expending energy to help get that stuff inside or outside of the cell. It depends on which way we're going. So in this case, what you see here is you see sodium ions. They're going into these carrier proteins here. And then this little thing here, this little like explosion, this is an ATP molecule. Okay, and you see the little ATP molecule there. The ATP molecule is basically breaking apart. Okay, and it's breaking apart provides the energy that basically opens up the other side of this carrier protein. So the stuff comes in this side, it can't get through, energy is expended, it causes it to change its shape. You see how it's kind of locked in there with that little part of the ATP, okay, the phosphate part of it, and then that allows more sodiums to go outside of the solution, okay? And then you also see that it's coming this way. What happens here? The potassiums, naturally, they want to come in this side, and so they're going to come in. You're going to see that phosphate come back out. And we'll talk about the ATP and all of that stuff. Basically, I mean, you guys probably remember from middle school, ATP is kind of like cellular money. This is where it gets the energy to get all this stuff to happen. So that's the active transport there. Active transport has to happen when you want stuff to go against what would normally happen with diffusion, when it goes against the concentration gradient. When I've got a bunch of stuff already out there, but I want to put more stuff out there, that is what active transport is. All right, so one last quick little thing here. Is we want to talk real briefly about what happens when particles are just too big. What happens when they're too big to fit through one of those channels? Because that happens. There are a lot of really big molecules that are unable to get in. So the way that happens is through two processes called endo and exocytosis is how they're going to get through the cell membrane then. So endocytosis is anytime we're going inside of the cell. Whenever you see the prefix endo, endo means inside. So like human beings, we have an endoskeleton. Our skeleton is inside of our body. Insects have exoskeletons. They've got stuff on the outside of their body. So endo means we're trying to bring stuff into the cell. And so that happens in a variety of processes, and it's not really necessary that you have to know what each one of these is individually, but we've got a process called phagocytosis, which is basically you got some big solid particle. The plasma membrane you see here sort of wraps itself around it, and then essentially it's going to pinch itself off, and you're going to have what's called a phagosome inside of the cell. And so that could be maybe some food particles. A classic example of phagocytosis would be like an amoeba, which is a single-celled organism wrapping itself around its food. it pulls it in. It pulls it into this little phagosome here. Okay, and then it's going to, a bunch of enzymes are going to go in there and attack that and break the stuff that's inside of there down. We've got penocytosis, which is where it's going to, instead of a big sort of wrapping around it, there's little areas that allow for particles to come in there, and then they're just going to come together really gently there. That's going to break off into a vesicle. Okay, we're going to see vesicles appear again here in a minute. Or you see that's got this sort of complicated term here, this receptor-mediated endocytosis. Don't worry about that terminology. That, again, just means that there's some chemical things going on. Something latches on and says, hey, you need to grab a hold of this. The plasma membrane basically wraps around it, pulls it in, and it becomes in a little vesicle here. Now, you see it's a coated protein, and we'll talk a little bit more about that later in the year. In exocytosis, it's essentially the opposite process. Instead of something the membrane wrapping around and pinching off to allow something to go into the cell, it's the opposite happening. And so in this case, what happens is the Golgi apparatus or the Golgi complex is going to wrap up a package, okay, and it's going to pinch that off, form it into a vesicle. That vesicle is going to then come up to the cell membrane, okay, and from the cell membrane then it's going to basically merge. The outside of the vesicle, remember, is essentially the same phospholipid bilayer that the cell membrane is. And so when it comes up to it, essentially just merges with it, becomes part of it, and then it's going to sort of just pop open and spit the stuff out. Okay, and so like that's what's the example of what's happening in both of these cases here. We've got a bunch of stuff here. There's a new membrane here that the Golgi complex made. It's going to move till it hits the edge of the cell, and then when it hits the edge of the cell, the membrane here that was in the package for the vesicle is just going to merge with the cell membrane, and the stuff that was inside of it is just going to get pushed out of the cell. Okay? So that's exocytosis. Hopefully that helps everyone. I know that there was some of that stuff that we covered in the previous videos, but that's a good summary of all the stuff that's in transport. So as always, make sure you make your flashcards, maybe pull some of these photos in and make sure that you know all the terms, and we'll do more work with the concepts in class.